Okay, hi everyone. I'm Rishidev Chaudhuri, and I'm going to be telling you about some collaborative work with a group with Tim Hanks, who's this useful gentleman here, in this group. We've got a couple of preprints out. The most relevant one is the top one, the biological one, and there are a couple of SFN posters on this material that I wanted to advertise. Okay, so kind of building off of Tim's talk, and working on the area in this talk, we're talking about perceptual decision making, which is this process where animals and humans integrate noisy sensory evidence and convert it into a discrete motor decision. So there's this classic example of the so-called random dots task, where monkeys or humans look at a bunch of dots on a screen. The dots are moving around somewhat randomly, but there's a bigger fraction either to the left or the right. And the monkeys have to look for a while, figure out what that is, and report. Here's a rodent example. These rats are listening to two speakers playing clicks left and right. They have to listen to the clicks and decide which side is now. So they integrate evidence and they convert it into some sort of motor decision. Perceptual decision making relies on a distributed network of cortical and subcortical areas. This is a rodent view picture, but there's frontal cortex, parietal cortex, the basal ganglia, thalamic networks, superior colliculus, and so on. A number of overlapping areas from Tim's talk. And one of the appealing features is that this network seems to be evolutionarily conserved across primates and rodents. They're homologous. Okay, so this has been for many years a rich paradigm for understanding how neurodynamics do cognitive stuff. Also, the citations are where the figures are from. They're not the people who came up with these ideas, the developers. So, for example, people have built these center diffusion type models where you integrate evidence to a bound, or people have built these sort of attractor network models where you argue that networks of neurons create these fixed point attractors in their dynamics and use those to improve the decision. So it's been a very rich and fruitful paradigm. Okay, so there's some basic questions you might ask about computation by dynamics in this network that I and other people are interested in. And so, for example, you might ask, what dynamical regime is the network operating? Like, what are the dynamics doing? They could, as I said, be forming fixed point attractors and using that to encode a decision. They could be a primarily stimulus-driven response. So you have these long stimulus transients, maybe low dimensional, maybe high dimensional, as in a reservoir network. Or maybe you have internally generated sequences of activity in the network that are somehow encoding information as these sequences speak through time. The last is close to what we think is happening in our task network. So you'll see more of that. You can also ask questions about how cortical and subcortical areas are really interacting in this network. Is the cortex really acting as a controller of integration by subcortical areas? Is cortex doing all the hard work and then telling subcortex, go ahead and actually execute the decision? Do we have some parallel loops, a fast loop through subcortical areas, a slow loop through subcortical areas that are sort of mutually coupled? So there are various possibilities for how areas can be interacted. And then I think one of the remarkable things about these networks and brains in general, they can be both robust and flexible. They can flexibly change to make decisions in different contexts. But there's been this very interesting series of perturbation experiments over the last decade, kind of inactivating areas we thought were important for these decisions and having much smaller effects than we would have. And so somehow this distributed network is also pretty robust. You can take out a node in the network and it seems to keep them. So I'm going to touch on, these are big questions. I'm going to touch on them in the context of our task and our analyses, and not going to give you a full answer, but I'm hopefully going to say something interesting about each of these three topics. Okay, so these are the questions we're asking, and we're going to do it in the context of an integration of evidence task developed and used in Tim's lab. These are rats. They're listening to a stream of sounds that are drawn from a Poisson distribution with some frequency. And at some point, the frequency increases. And so you see click, 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 and at some point it gets faster and goes click, 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 click. And the rat has to detect that change, pull its nose out and go get its root. And so it's a change detection task. And they need to make, decision within some time window. Otherwise, they get nothing. This is just to tell you that the task sort of works. Animals do it. They do it faster when it's easier, and they seem to be responding to evidence. I'll skip over methods a little bit just because it's a short talk, but I'm happy to slow down and talk about any methods you like. Okay, and then Tim's group are using NeuroPixels probes to record right now from 4 areas of this network. And we're focusing here on results from an area in frontal cortex, the so-called frontal-oriented fields, and an area in the basal ganglia called the pterodorsal striatum. And we think we know that both of these seem to be important for the task. So we have these population recordings, on the order of 50 to 200 neurons per brain region, depending on the session. And so it is a pretty rich data set. Okay, so the first thing we find, and I'm just going to kind of run you through some of our conclusions, is that, so this is a dimensionality reduced picture of activity in both the striatum and frontal cortex as animals do the task. Over here, this is while they're integrating evidence. They start blue, go to red. And you find these kind of smooth, relatively stereotyped trajectories, pretty dynamic. It's changing over time. It's not a fixed point, but pretty stereotyped. The dark colors are showing you the mean, and the light colors are showing you the scatter across trials. So this is a dimensionality reduction projection that's trying to pull out the structure over time. And so it's pressing another step. But we find these trajectories through time. And one interesting thing is that they look very, very similar across both these areas, cortex and striatum. And that's a theme we see again and again. Cortex and striatum seem to be doing very similar things in a lot of them. The strongest signal we see in these data, as you might guess from those pictures, is time. So these are both kind of confusion matrices from decoders trained just to pick up time. We took the simplest decoders we could. This is just linear discriminant analysis. So we're saying, take a simple decoder, can you put a hyperplane in and decode the moment of time in the task you want. And these confusion matrices tell you basically you should be looking for a bright band along the diagonal. I don't mix up time 1 with time 5. And that's what you see. And again, that holds across sessions. Now the color traces are showing the average decoding error across time, striatum and cortex, and the light traces are showing the scatter across trials. Strong time signature. You can then go in and you can group neurons by the decoder coefficient to say how are these neurons encoding time. If you group them by the first mode, as you probably expect, you see these smooth ramps. If you group them by the second mode, you see these bump-like transient dynamics. And you can group them by both modes. And I think the theoretical contrast to draw here is between this kind of smooth time encoding and more of a sparse code. So we don't see a sparse sequence code where this group is active in that group, and that group, and that group. It seems comparatively dense, but moving through neural state space. We did some work to characterize the geometry of these trajectories over time. And so that's an illustration of neural state space. We're plotting trajectories in this n-dimensional space where each axis is a neuron. Our general analytical approach has been to take a locally linear approach. These things are nonlinear objects. And so we don't want to fit some linear model to the whole thing. But since they're smooth, we can look at little pieces that fit a linear model to each. And so over here, we're measuring the dimensionality of these trajectories over time using this locally linear approach. And we find that the dimensionality remains pretty stable over the evidence evaluation period, that's the left plot, and then moves into a lower dimensional subspace when the animal makes the decision for the movement. Okay, so a few slides of differences between the areas. So far, I've really highlighted we have this dynamic sequential coding, but it looks the same between these two areas. And I haven't shown you correlations, but the areas are just as correlated with each other as they are with themselves. If you line things up to the movement is now time 0 in red. You start to see signatures of a difference in what frontal cortex is doing. The trajectories seem to kind of accelerate and get more spaced out in frontal cortex. Again, we went back to the geometry of the trajectories and asked, how do the the subspaces that capture the variance of trajectories, how do those change over time? And so if those curvy lines are trajectories from different trials, there's some linear subspace that captures what happens early in evidence evaluation, late in evidence evaluation, and movement. And we were inspired by work in motor cortex to construct some sort of alignment index that captures how these subspaces change. I can go into the method a little bit more, but basically what we do is we use PCA to find subspaces of high variance during one task effort. And then we say, how well do those subspaces of high variance capture activity during another task effort? And so those are those two plots there. If the subspaces are very different from each other, there should be a big gap. My PCs don't capture your variance. If it's stable, there should be a small gap. My PCs do capture your variance. And so we do that, and we find if you look, as you transition from evidence evaluation to decision, you see that frontal cortex really seems to lead. The striatal subspace remains comparatively stable, small gap. The frontal cortex tends to rotate, bigger gap. And this holds up across sections. Everything is below, most stuff is below the diagonal, showing that frontal cortex seems to play more of a role in the decision commitment, in line with prior work. One slide on, optogenetic perturbation. They confirm this sort of thing by going in optogenetically and activating frontal cortex either early in the trial or when the animal is about to make its response. You find that if you do it late in the trial, it seems like the animal makes false alarms. And so it really seems like frontal cortex is kind of keeping things in place. But you only need it late in the trial. Early in the trial, you can recover from having it inactivated, but late in the trial, you're much more sensitive. If you want to know more about that, Kendall has a poster on the system. Okay, so this is a bit of a whirlwind of our results, but so far, I've told you that what we see in this task is that the network seems to be following these stereotyped nonlinear paths, suggesting coding by these internally generated neural trajectories, rather than like fixed point attractors or low-dimensional stable manifolds, and also not like a very stimulus-driven, like a reservoir network or a low-dimensional stimulus-driven. We also see that, rather than cortex and subcortex doing very different things, it looks like the basic unit in our tasks is this flexibly modulated corticostriatal loop. They're mostly pretty tightly coupled, but cortex seems to be taking the lead in deciding when you actually make the decision. Okay, so I'll touch on the last topic a little bit. And to that, we kind of shifted gears, and one of the interesting things we see in our data, and a lot of people see just experiments, is that animals seem to switch between different states. The same way that listening to a talk before lunch is different from listening to a talk after lunch. If you look at what these animals are doing, they'll have clusters of hits, they go, 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 and then they totally space out, they're checking their phones, they're grooming, they're kind of wandering around, and then they kind of come back in and engage. And so we decided to capture these states and then try and ask how the decision-making circuit behaves. And so to do this, we adapted work developed by, Kalun and Zoe Ashwood, people in Jonathan Fiddle's group. And what they do is they fit a latent variable model that tries to capture these different states. And these models basically assume that your animals transitions between some small number of latent states, things like task engagement, arousal, hunger, and that these latent states shape the relationship between task inputs and task outputs. How much am I paying attention, how responsive to evidence am I, and so on. So we just really adapted their workflow with shared really accessible code online. Okay, so we find that in our task, these rats alternate between two states that we call engaged and impulsive for reasons you'll see in a moment. That's a plot of kind of posterior state probability And you see that, they're in engaged state, and then they switch, and then in an impulsive state. In this session, they're more engaged than impulsive. That's always true. The model captured, we can look at things like what are the probabilities of different outcomes in these states. That's what we actually fit. And as you'd expect, the impulsive state, lots of false alarms, they respond too soon. In the engaged state, they're more hex, they stay on task. They're still pretty false alarm prone, at least in this set of sessions. Okay, so we're just exploring this, but one of the interesting things we find is that if you look at these different states and you say, let's look at the firing rates of neurons in impulsive versus engaged, you don't really seem to see a difference. Stuff lies along the y equals x line. We don't see a strong effect on firing rates. Where we do see an effect is on this corticostriatal coupling. And so the x-axis I'm plotting the engagement probability that we get from our Vicky variable model. It says 80% chance this was an engaged trial. On the y-axis, I'm plotting a measure of frontostriatal coupling, just the average correlation between pairs of neurons. And you see this kind of like nice linear increase. And so when you're more engaged, there's more sort of frontostriatal coupling. If you'd like to hear more about that, I was giving a presentation poster in the same session, the same group. So, okay, so to wrap up, I told you that at least in our task in our hands, we have this picture of sequence coding. Activity seems to follow these sequential paths. Our thinking has been, very influenced by this really nice paper from Suan Kuei from David Tang's lab, where I think they articulate very nicely what this kind of coding picture looks like. Stereotype sequences through time with information multiplicatively encoded around that. That's been very influential on how we think about these data. We see corticostriatal loops, maybe not a surprise to people who study anatomy, but it really doesn't seem like we're going through sequential cortexes, this, then striatum does that. Rats seem to switch between two states with corresponding changes in frontostriatal coupling. And as my talk title says, what we're seeing is a highly dynamic and distributed decision-making circuit that's strongly shaped by internal state. Okay, so obviously I thank all the people who worked on this project, including I who did the latent variable modeling, had the poster, and indeed I did some of the more dynamic dimensionality modeling. Daniel developed a bunch of the recording methodology, as did Adam. Again, did the opto, and again, as a poster, and Preetham did some of the other experiments. I also wanted to mention Katie, even though she didn't work on this project, because she has a poster and she's looking for postdocs. So if you want to hear about the curvature of neural manifolds, please go to her poster on the 19th. I'd like to thank funding from the NIH and especially the NSF CRCNS, which really sort of helped kind of launch this collaboration and helped it go in a lot of different directions. Yeah, so thank you for your time.
We can activate two-phase possible.
Yeah, so the question was, when we inactivate FOF, do we see an increase in false alarms? And so that says, yes, with some caveats, this is somewhat preliminary work. And I guess the second part of the question was, this seems to be opposite what you'd expect from, just like a motor structure. This task has a little bit of a flavor of a go, no go task. The animals are kind of sticking their nose in, they're listening to these clicks. They can be pretty false alarm prone. And so it seems like one of the things, and again, I'm speaking as a theorist who doesn't do these experiments, it seems like one of the things that's hard for them is just to kind of stay engaged, like stay there and wait. And so it seems like maybe FOF is like helping with whole decision. Like maybe that's the dominant effect. But don't quote me too much on this. This might change over time and the theorist. But that's my interpretation. Those certainly some more activation results that are a little bit more complicated. So yeah. Let me Dr. Campbell if you can.
Have we checked whether the pulse around inclusive states are we pulse around that are inclusive in the sense of relative continuous, or whether the element have the lower the threshold and in fact what it in constant burst of peace in the baseline stimulus?
So I'll repeat the question to you. That's a great question. So we have these impulsive states, and one of the ways we're measuring impulsive is we're looking at false alarms. They kind of react too soon. And the question was, but are those false alarms actually evidence-driven? It's a random stimulus. Sometimes it fluctuates up. And if they are evidence-driven, is it using a different, like kind of a lower threshold? Or is it just like it's not missing a stimulus or all kind of works out? That's a great question. We're doing that at the moment, and it's sort of so you can basically look at these like reverse correlation kernels. It's sort of I don't feel like I have a clear enough picture to sort of say one way or the other. I'll say they do seem like they're sort of responding to evidence, but I don't know if it's kind of a lower threshold. But it's a great question.